Good morning, students. Welcome in today's science class. Today we are going to learn first lesson, Living World and Classification of Microbes. Biodiversity and need of classification. Last year, we learned that all the living organisms on Earth have adopted according to the geographic regions, food ingestion, defense, etc. While adopting, many differences are observed in the organisms of same species too. According to 2011 census, around 87 million species of living organisms are found on the earth, including land and sea. To study such a vast number, it was essential to divide them into groups. So groups and subgroups are created considering the similarities and differences among the living organisms. This process of dividing living organisms into groups and subgroups is called biological classification. Robert Harding Whittaker, 1920 to 1980, was an American ecologist. In 1969, he divided living organisms into 5 groups. For this classification, Whittaker considered following criteria. First, complexity of cell structure, whether the cell is a prokaryotic or a eukaryotic. Second, complexity of organisms, whether the animal is a unicellular or a multicellular. Third, mode of nutrition. Now in plants, there is autotrophic and photosynthetic nutrition, whereas fungi are saprophytic, absorption from dead organisms, and in animals they may be heterotrophic and ingestive type. Fourth is a lifestyle. Plants, they are producers. Animals are consumers. Fungi is decomposers. Now fifth group is phylogenetic relationship. The prokaryotic to eukaryotic, unicellular to multicellular. Now we will see the classification in the flow chart. Living organisms divided into 2, prokaryotes and eukaryotes. Prokaryotes further divided it into unicellular, whereas eukaryotes divide into 2, that is unicellular and multicellular. Unicellular is further divided into kingdom Monera. Now, in eukaryotes, the unicellular is divided into kingdom Protista, and multicellular divided into 3 kingdoms, that is kingdom Fungi, kingdom Plantae, and kingdom Animalia. Now, in kingdom Fungi, cell wall is present, but organisms can't perform photosynthesis. In kingdom Plantae, cell wall is present and organisms can perform photosynthesis. Kingdom fifth, that is Animalia, no cell wall is present. Kingdom first, Monera. In this, we have to do one activity. Take a small drop of curd or a buttermilk on a clean glass slide, dilute it with a little water, and carefully keep a cover slip and observe it under high power of compound microscope. What did you see? Moving small rod-like microbes are lactobacilli bacteria. All type of bacteria and blue-green algae are included in the kingdom Monera. Now we will learn the characteristics of this Monera. First, all the organisms are unicellular. Uni means one, having one cell. So the organisms having one cell are called unicellular. Second, they may be autotrophic or heterotrophic. Means they can produce their own food or they may depend on somebody. Third, these are prokaryotic cells without distinct nucleus or cell organelles. Kingdom second, Protista. Activity: prepare a temporary mount of 1 drop of pond water on a glass slide. Observe it under low power and high power of microscope. You will find some motile microbes with irregular shape. These are amoebae. Now we will see the characteristics of Protista. First, Protista are single-celled organisms with well-defined nucleus enclosed in a nuclear membrane. Second, they have pseudopodia or hair-like cilia or whip-like flagella for locomotion. Locomotion means for movement. Pseudopodia. What is meant by pseudo? Pseudo means false. Podia means legs. So false legs. That is called pseudopodia. Third, autotrophs. Example, Euglena, Volvox. They contain chloroplast. Heterotrophs, example, amoeba and paramecium. Kingdom third, fungi. Activity: take a moist piece of bread or bhakri and keep it in a container with lid for 2-3 days. After 2-3 days, a fine cotton thread-like tuft is found growing on the surface of the bread. Prepare a temporary mount of few threads from this culture and observe it under the microscope. Now we'll see the characteristics of this. First, these are non-green eukaryotic heterotrophic organisms. Non-green, they don't have eukaryotic, they don't have distinct nucleus. Heterotrophic means they grow on others' body. Second, most of them are saprotrophs. They feed upon decaying organic matter. Saprotrophs means they grow on decaying organic matter. Third, their cell wall is made up of tough and complex sugar called chitin. Fourth, some fungi are thread-like and many nuclei are present in the cytoplasm. Fifth, examples are baker's yeast, aspergillus, fungus on corn, and penicillium mushrooms. Though many systems of classification are introduced after the Whittaker's, his 5-kingdom system is widely accepted. I hope you have understood today's lesson. We will meet in next session. Thank you.